Hello, everyone. Thank you for joining us today. My name is Dr. Jeremy Petrovich of Wiley's Current Protocols. I'll be assisting today in moderating our Ask the Experts panel on smarter and simpler flow cytometry panel design, sponsored by BD Biosciences. I'd like to introduce our panelists today. First, we have Bob Balderas, VP of Biological Sciences and VP of Marketing at BD Biosciences, who is a world-renowned expert in flow cytometry. Thank you for joining us today, Bob. Next is Dr. Yolanda Mahnke, founder of FlowKnowHow LLC, whose influential research career has led to numerous advances in flow cytometry techniques. Next is Dr. Gardner, head of the flow cytometry core facility at Memorial Sloan Kettering Cancer Center, whose expertise in math, physics, and biology brings a unique perspective to flow cytometry. Lastly, with us is Dr. Mirko Corselli, Global Scientific Content Manager at BD Biosciences and leader in identifying cutting-edge technology for flow cytometry. Thank you all for joining us today, and we begin with Dr. Corselli providing an overview of today's discussion.
Thank you, Jeremy, and thank you everybody for joining us today. I'm really excited today to continue our discussion on panel design, and I'm particularly grateful and excited to have also a panel of experts that will provide us with their perspective on panel design and help us addressing some of the numerous questions that came through after the webinar. So before doing that, we would like to go briefly through a summary of what was discussed at the webinar, and that introduce the questions that will be addressed by the panel. Next slide, please. So just as a summary of what we discussed at the previous webinar, we discussed how several factors contribute to the complexity of panel design for multi-parameter flow cytometry, including the biology, the fluorochromes, and the instrument. We specifically focused on spillover and spread as one of the major factors that affect panel design. And we also talk about how the higher capability instruments allow for use of new dyes with less spillover. And eventually, we showed how this can simplify panel design while allowing selection of fluorochromes that have minimal impact with each other. We also emphasize the panel design rules still need to be applied, even though we do a selection of fluorochromes with minimal challenges. So it's really critical to still understand the panel design rules. We also emphasize how variability can occur from instrument to instrument depending on different configurations. So how critical it would be to understand the instrument you're using. Next slide. One of the central themes of the webinar was the use of a spread matrix as a reference to identify fluorochrome combinations that might be challenging in terms of spillover and spread. We talk about the caveats of using this spread matrix and how the amount of spread would be eventually variable depending on the biology or the instrument configuration. But overall, we can use a tool like this to identify challenging fluoro combination and focus on combination of colors that have minimal impact with each other, like the one in the table below, where in this case, we were able to select 10 fluorochromes with very minimal spread and spillover, which simplify the panel design and ensure high quality data. Next slide, please. Another critical point that we emphasize is that the number of fluorochromes or foundation colors that can have minimal impact is a function of the complexity of the instrument. So if you look at a 3-laser instrument, based on the real estate and the space that we have available, we could find 6 colors that work very well together with minimal challenges. But if we move to a 5-laser instrument with 18 parameters, now we have more room to physically separate the fluorochromes, and we can go up to 10 foundation colors. And use a critical point to emphasize that larger instruments are not only valuable to design large panels, 14, 16 parameters, but also can be very useful to simplify the panel design of smaller panel in the 8 to 12 realm. Next slide. We also emphasize that, again, spillover was just one or the main factors influencing panel design, but there are a number of other considerations to be taken account, like the biology, the fluorochromes, and the instrument. We will receive over 200 questions, very engaged conversation that really touch upon all the other aspects of panel design. And that's why we decide to continue our commitment to you to provide information and tool to understand the intricacy of panel design. And rather than do this again with just one perspective coming from BD, we really decided to bring in world-renowned experts that can bring their perspective and address more generally all the aspects of panel design and address most of the questions that you have sent to us. And so with that, I would like to now pass the discussion to the panel. And with the next slide, we have selected a couple of questions that really encompass the majority of questions. And we'll go through a series of questions that are going to be addressed to our panelists before opening up a live Q&A question session to address questions that will come live as we discuss. So I'd like to pass to Bob Balderas now and start with the broad questions for the panel, which is, what is your step-by-step approach to panel design?
First of all, thank you, Mirko, and thanks for just to revisit some of the concepts and issues that you addressed several weeks ago. Like everybody, there are multiple ways to begin with a panel, but just to make sure that we highlight one more time, we learn more and more when we say it over and over again. But I think for each of us, we address multiple things. We need data, and we use data-driven approaches, which actually even reduces the amount of time and the cost of everything that we do when we're building a panel. But we need to take always we always need to look at the biological tree. We examine receptor densities for each of the different receptors for the surface. We look at the amount of protein and try to determine that if it's an intracellular one, because we can't get an accurate amount of the protein like we can on a receptor. We have to look at the colors available on an instrument and the configuration that's there, but always do we have the ability to change the filters, in the instruments that we have. We'll review those spread matrix that have been generated on a given instrument, and then we always examine a couple other things, and that is that each laser line there's a story. And for many people, that story is still something that we're not all familiar with. But we all know that the very first color off every laser is a real good one, and they usually don't spread too much into different channels or cross lasers. But there's also a story about the other end of the color at the other end of each laser line. Those are also very good colors, and they don't spread a lot. And when we look and try to get all that information together, it allows us to look and build a panel in a different way. So, because at the end of the day, it's wherever you start, well, that's where the journey begins. And so for myself, the way I look at a panel is I address it by looking at the laser lines for whatever instruments that I have, and I break those up into 3 parts. The first part is that I address my first specificities, meaning at the top of my biological tree, the more gating let's say receptors, a CD and a T cell, the CD3s and 4s and 8s. And I build those with the colors at the very end of each laser line. They're good colors. They're not the brightest colors, but again, they don't spread very much. And as soon as I tag each one of those specificities to a given color, then I move all the way to the short wavelengths. So I move from the long wavelengths, and my second step is to take advantage of those ones that are really good, right? They're relatively bright out there. I probably, in my first steps, got rid of the very high expressing receptors or in my panel. And now I'm in the middle of my panel in terms of receptor densities, let's say. So I'll use the very short wavelengths off every single laser line. And then for the last third of the panel, well, that's that valley of doom. That's in that area of the 600 range where there's a lot of spread and a lot of spill. But we know our rules, and those rules are that towards the end, towards the bottom of our panel, many times, those are specificities that are not expressed at very, very high levels. And because of the fact that the receptor density or the protein amounts are relatively small, then by putting those in that range of around 600, there's not enough color to cause enormous problems with the spill and the spread that we see with conventional flow. So that's the 3 steps I take in building a panel. Rui, maybe you can bring another perspective on there or Yolanda, maybe you can bring another perspective for how you address a panel.
Thank you, Bob, and thank you also to PD and Wiley for having me on this discussion. I like your approach very much, and I also agree with what Mirko said. You have to know what you're working with, and that's at the level of the biology of your samples, as well as the instrument and the fluorochromes, whatever reagents you use to interrogate your samples. I always like to lead with the biology. So what are you trying to address with your panel? What are you trying to find out? What are the important markers? So really define what it is that you want to measure, which markers are necessary to do that, which are important, and which are just plain interesting to add if possible. So you kind of add a little ranking into the markers that you might want to address. And if you have knowledge about co-expression or mutually exclusive markers, that's something good to note. You don't always have that information. Especially if you're in a new field or looking at markers that are known but in a cell subset that hasn't been studied for those. And then I like to look at the expression density and expression patterns of markers along the lines of what Bob just said. So I like to kind of bin my antigens into primary, secondary, and tertiary, where the primary antigens are going to be very well characterized and typically are on and off kind of markers like CD3, either the cell expresses it or not. There's not much in between. And they're usually markers that are used for categorizing broad subsets. And then the secondary antigens are also well characterized and expressed at relatively high molecular densities per cell. But there's usually a continuum of expression. So you'll have kind of a smear going from negative all the way to high expression. Something like CD27 or CD45 RA would fall into that category. And finally, you have the tertiary antigens, which are low expression markers. They don't always separate well from the negatives. So the thresholding is not always very clear. And of course, your uncharacterized antigens. So if you're looking at something new, you just don't have information about that. So I would typically, the first antigens that I would try to assign reagents to, or fluorochromes to, are the tertiary ones, because oftentimes there you don't have a lot of choice. You might only have 1 or 2 fluorochromes that are available, so that's what you want to start with. And then add in the other ones, working with the knowledge that you have of your instrument performance, so working with a spillover spreading matrix or other, there are multiple similar types of matrices that are being used, as Mirko just showed in the previous slides. And then it's very important to always titrate your reagents to really optimize the panel, and that includes the reagents and the way you use them. And we can talk a little bit more about that if you want. Of note, if you have a good collection of antibodies in your lab and they're already titrated, but there are different instruments that you could use, just make sure that the titration actually works for the instrument that you're wanting to work on, because the optimal titer can differ from instrument to instrument, particularly if there are different lasers involved. So, for example, something like a Cy7PE will look different on an instrument that uses a green laser versus one that uses a blue laser to excite that fluorochrome. And of course, the titer might also depend on the cell type you're using. So there are different considerations you want to take into account. So just because it's been titrated once doesn't mean that it works for your particular application. And then you want to really test your panel, which doesn't just mean, throw all the antigens, all the reagents together that you picked out and see if it looks good. But, do kind of elaborate FMO experiments where you leave out each reagent in subsequent samples to see if you have some spillover spreading that really affects detection of some of your antigens. Try some variations of the panel to see if you can optimize and improve detection of some of your weaker expressed antigens, etc. And then finally, you want to validate the panel. And there are different types of validation that you can do. It depends how many people, for example, would be involved in that research. So you definitely want to do intra and inter-assay variation testing. And if there are multiple people, you also want to do inter-operator testing. And again, coming back a little bit to just working with the biology and not taking for granted that a reagent works in your system that worked perfectly in somebody else's. CD4, of course, is very highly downregulated in HIV. So just because it's a bright marker in most situations and you don't have to worry too much about how to stain it, if you have severe HIV infection, then detecting those CD4 molecules can be quite challenging, and you might want to have to stain it intracellularly. So there, just think about what might be particular in your system. If you're using specific buffers that might affect markers of fluorochrome, etc., and whether you can circumvent that or whether you might just have to work with other markers instead. So that's just kind of my approach in a nutshell. Of course, there's always more to it than that, but I'll let Rui pick up from here and see what else he has to add.
Okay, thank you. Well, I want to also start by thanking everyone for the opportunity to be here, trying to address these such important questions. I disagree completely with what Yolanda said.
Of course.
I'm joking. No, it's. I just want to add, or rather emphasize, what you were just talking about. I think it's very important for people to understand that panel design is not just a theoretical exercise where you try to identify the best combination of colors. There's a lot to do regarding validating the panel. First, understanding if your instrument is well QC'd or not. So you need to have a very well, an instrument performing very well. And then there's all of these steps of titration, validating your panel, looking at spread, using the right controls like FMOs to troubleshoot. So there's a lot of work there. And again, I mentioned this because I see a lot of researchers getting frustrated with, okay, I have a panel, no, it should work, right? I should just run it and everything is going to work fine. But it's not. It takes a big amount of time to validate all of these fluorochromes. And then actually there are a lot of steps where we need to change the panel, and it's just an iterative process until we find the right panel. So I think that's an important aspect that you mentioned, Yolanda. that people should understand that they have to go through all this process, and it takes time. It takes time.
So the next question, and Yolanda, why don't you begin with this one, is what are the most important controls to ensure reliable, high-quality data?
Yes, so I think we briefly touched upon that already. You do need to, like we said, interrogate your panel at every step and make sure you have the right controls. Also, once the panel is done. So when you run your experimental samples, you want to make sure that it's still giving you the answers that you're looking for, and that you can make statements of what's going on in your experiment. So, of course, single stain controls are indispensable. So, how else are you going to come up with a good compensation? So, you will all know these are either cells or beads that you stain with one of the reagents only, and they should really be the exact reagents that you're using in your experiment. For something like FITSI, it doesn't matter because the spectrum that it emits is a physical property of the FITSI molecule. But specifically for tandems, you really have to use that exact reagent for your compensation controls. And here I just want to add a note of caution where it's like P-Cy7, everybody can see that it's a tandem. It's in the name, there's no question there. But a lot of the newer reagents, let's say, they kind of hide their identity. So they just have one short name and you don't necessarily know if it's a tandem or not. So just as a good practice, just use the actual reagent that you use on your cells, on your experimental samples to stain your compensation controls. Then there are FMOs, so the full panel -1 that I just briefly mentioned already as well. So these are samples where you stain with your full panel minus one of the reagents, and you don't necessarily have to do that for every single reagent in the panel once you're done with the panel design. But you might want to include it for one or two markers that are very hard to detect, just to kind of, if it's hard to pick out from the background noise, it might give you some indication of if you have a staining or not. Basically, the FMO allows you to determine whether there's a lot of background noise due to spillover spreading from other reagents that are in there. But as a cautionary note, you should not rely on it for setting gates, because the threshold of where your gate to discriminate between positive and negatives should be set. The FMO can't really tell you that because the negatives are usually also affected by adding in that extra test antibody. Sorry. So the threshold might be a little higher than your FMO would indicate. And so people like to use isotype controls, which I would not recommend at all. Like, again, all of those controls have their place, but as we mentioned, you need to really understand what every reagent or every technique or every filter does for you. So isotype controls, as the name says, they have the same isotype as the antibody that you're controlling for, with a different specificity, so they shouldn't specifically bind to anything in your sample. And they use the same fluorophore. Now, if they don't have the same fluorophore to protein ratio, though, to antibody ratio, as your antibody that you're controlling for, it's really of not of much use. And typically, you don't have a good match of the fluorochrome to protein ratio between the isotype control and your test antibody, let's call it. So, in that sense, it's really not going to help you much, and you will have to use it at the same concentration. So I've seen cases where the isotype staining gave more of a background than the antibody that you're using. So just be very cautious with those, and again, don't use those for setting your gates. The best are biological controls, and I realize they're not always possible, but oftentimes, particularly if you have a mixed biological sample like PBMCs, you usually have a cellular population that is negative for the marker of interest. So, and there you want to look at a population that is as similar as possible to the population where you're measuring the marker. So let's say Ki67, for example. I would go, if you're looking for proliferation in T cells, there are going to be few cells that are Ki67 positive. Look at other T cells that are Ki67 negative. They will give you a great threshold for setting that gate. They will be preferable to, say, B cells, because B cells and T cells are going to be stained with different reagents, so they will have different background noise coming from those reagents that are used to identify them. And then there are multiple biological controls that you might want to think of. Again, you know, are you using some buffers that might affect some of the stainings, or if you're doing activation assays, have a sample that has not been activated, as well as one that has been activated, so that you know you can actually measure all your antigens. So if you're looking for activation and everything is negative, it might be that none of your samples react to whatever you're testing, but you're not 100% certain that you actually added everything correctly, that everything's working correctly. So you want to have that positive control. So again, let biology drive your decisions here and know what you're working with.
So our next question, why don't we direct it towards you? What are the best practices for compensation, and what are the most common mistakes with compensation?
Right. That's a very important question, because we do see a lot that it's very hard to promote best practices in compensation. I would say so let's think of what compensation is, right? What are we trying to do? So essentially, we're trying to quantify the amount of spillover, right, of a certain fluorochrome in different channels. So if we're trying to quantify this spillover of a particular fluorochrome, then I would say the most essential best practice is to use the exact same fluorochrome, and Yolanda touched this exactly. You need to use exactly the same fluorochrome that you're going to be using in your sample, or else you're not going to be able to quantify correctly, right? So this I would say that the most common mistake is exactly not applying this best practice. It's so common for people. Well, it's obvious if you have GFP. Some people, they can't have just a cell line just expressing GFP, for instance, and then so they don't have that single stain control, and they'll use FITC, right? So just because it's green. This is not so common anymore. People realize that they need to use something similar. But here's the problem: is we don't want something similar. We want the exact same fluorochrome, and I'm repeating this on purpose because that's the most important aspect. We do have issues like tandem dyes, like Yolanda mentioned, because essentially, if you think of tandem dyes, as being something, the way they're manufactured, it's very difficult to assure that tandem dye is going to always have the same exact spectral properties, right? If the spectral properties will change from lot to lot of a tandem dye, the spectral properties will change if they're exposed to light, if they're out of the fridge. So even if you buy, a few vials of the same antibody conjugated to a fluorochrome, if you use one vial more and it's exposed more to light or being outside of the fridge, that tandem dye will start degrading, and the spectral properties of that tandem dye will change and can change significantly. So we had one scenario where somebody was running their samples, and we noticed that compensation wasn't working very well, and we asked, so did you use exactly the same antibodies from the same lot, antibody conjugate from the same lot? And the person said, yes, I used the same. And that got me wondering, but wait a minute, but did you use the same vial? And then they answered, no, we actually saved the new vial for the sample, and we used the old vial for compensation. And so just that was enough to throw off compensation and basically mess up the panel. So I think, thinking of these in these lines, that you need to use exactly the same vial to assure that you're using exactly the same fluorochrome, I think is the most important aspect. Then, of course, this is one of the rules of compensation. Then there are other rules that are commonly mislooked. Like, for instance, you need to make sure that the carrier of the fluorochrome that the antibody is attached to, that the negative and positive have exactly the same autofluorescence. Because when calculating spillover, you're basically subtracting that that fluorescence to make sure that those two negative and positive populations are equally negative to all of the secondary channels, right? So that's an important aspect. The other rule of compensation is that the compensation control should be as bright or brighter than what you see in the sample. And this is also something very easy to overlook. We see many times, typically, especially when people use beads and cells interchangeably, you're not seeing the same, let's say, expression, right? So it's very easy many times to realize that your beads maybe are not as bright or brighter than what you see in your sample. So that's another common mistake. So having those rules in mind and understanding that anything that you do when you're running your single state controls or compensation controls, anything you do, if you don't follow one or more of these rules, this is going to mess up compensation. And actually, you can think of it in the opposite way, which is if you do see any problems, it's because you missed one or more of these rules. So it's very important to have them in mind and apply them, I would even say, religiously, because you're always going to see some problems with compensation if you don't follow these rules. Now, this brings me to a question. I don't know if this is going to be addressed later or not, but we can talk more about that, which is the reason why people don't really feel that they need to follow these best practices is because, at the end of the run, you run all your samples and you see that compensation is not, correctly evaluated or calculated. What most people do is they tweak by hand so that the populations match and are nicely, in line with the same median. Now, this is probably the biggest mistake that anyone can do in compensation, because tweaking compensation, as the actual word tends to allude to, is manipulating data, right? You're basically using all of these controls so that you have compensation properly calculated, and then you go by hand and change things. So this is extremely negative for the quality of data that's produced. It impacts rigor and it impacts reproducibility, because when you look at a paper and you're looking at data and you realize that, wait, somebody here was just, at the end of the day, was just tweaking things and matching them, it's very hard to reproduce something like that. And it's also very easy to make mistakes when changing things by hand. So this is a very important aspect, because, again, thinking that you can tweak things at the end also deters you from applying the best practices in the first place, right? And again, if you see something wrong with compensation, it's because you didn't apply the proper compensation controls. So I think those would be the most, common problems in compensation.
All right. Thank you very much. This last question is directed towards Bob. How important is it to know antigen expression levels and co-expression patterns? And where can someone find this information?
Again, a really good question. I'd like to address perhaps one specific question before that, and it's about intracellular protein information as opposed to receptor density data on the surface of a cell. And I think for many of us, we do know that there are tools today and applications that will allow us, utilizing PE as a color, that allow us to be able to do some quantitative work on the surface of the cell to determine the numbers of antibody binding or antigen binding properties of a specific cell. And there are several different places in the world, for which that type of technology can be obtained. But I think one of the important things is when we go into the inside of a cell, that the quantitation technologies don't work as well. And they don't work as well as because we have to fix and perm our cells. And when we do that, we cross-link the proteins inside, and that is impacted by time and temperature and many, many different types of variables. So trying to get a real accurate count of the numbers of, actual epitopes inside a cell for a protein is actually quite difficult. So one of the things that you can do is simply take a PE conjugate of any one of those proteins in the inside of a cell and look at the MFI for a given protein. And that sometimes gives you a relevant, kind of an indicator of the amount of protein that may be there. To get back, though, to the question about receptor density, there are actually several sources for information today. Recently, there was a publication by Pablo Engel and his team in Frontiers of Immunology. I think that was volume 23. It was in October of 2019. That actually did a very good job of looking at CD1 through CD100 for human, both on blood, on bone marrow, and also in thymus. And so it's an excellent resource to be able to get the types of information for co-expressed different receptors from 1 to 100, as well as to get the relative frequency that can be on additional subsets of cells. Now, in addition to that, there is unpublished work that we have at BD that was done several years ago in a collaboration with Peter Krutzik's company called Primity Bio, now part of Caprion. And that database, we have made it available for individuals just by asking in BD, and we're more than happy to provide it for you. It does explore approximately 380 CDs. It looks at receptor density, the relative amount of PE that's expressed. We look at about 35 different subsets of cells. So the database can also help you and determine what is actually expressed on what cell and or what subset of cells. And that's very, very valuable information as it relates to, building looking at the biology and also assigning colors. The third source is a new source, and this is for all the mouse immunologists in the world. Just recently, with work that we did at BD in a project that was in collaboration with CIF, which is a company out of Marseille. I worked with Hervé Luge, also Bernard Mallison and Marie Mallison and their team, and we built a similar model for mice. We took one mouse strain, but we did the work a little bit different. We borrowed the knowledge from everything we did from the human perspective. We examined all the different cell surface receptors antibodies that were available across multiple companies. And what we did is we looked at basal spleen. In addition to that, we stimulated the splenocytes with anti-CD3 and then looked at receptor expression and then subsetting, so we know what's on what cell type. And in addition to that, we injected the mice with poly-IC, a TLR9 agonist, and looked at an inflammatory response. And we have produced data on all three of those areas. That data is being combined right now for publication. We have spearheaded initial thoughts and how we're going to write that specific paper. If there's any need for any specific information right now that would help anyone's, panel design, I'm more than happy to help you and give you the actual numbers out of the database. It's not ready at this moment to be out there for everyone to have, but it will be part of a published work that we are working on right now. I think those are the three probably best sources of information today.
All right, great. Thank you very much, everyone, for those insightful answers. At this time, we are going to move into the live Q&A session. And our first question is directed towards you, Bob. Someone is asking what specifically you were referring to when you mentioned end of the laser line. Okay,
well, that's good. It's a good question, and address this in other webinars. But what we do on different instruments is we use an antibody like CD4, and we test the entire instrument with every color that's possible that can be run off of any given instrument. So let's just pick one like like a UV line, the UV 355 line. So there's a specific number of colors off each one, and the first color off that would be BUV 395. So that's the first color off that laser line. The last one in the laser line is all the way out in the 800 range. So what happens is we test the anti-CD4 with every color, and we open up every single detector, and we look for that spill and spread into every open detector. So on a given specific individual laser line, what we do is you can look at it at that color, let's say BUV 395, in every single channel or every detector on the 355 laser, and look to see the amount of spread. This is how we generate the spread matrix. Likewise, and what you would see then with 395 is there would be very, very little spread into the other detectors. If you then go to the last 800 color that we develop, and then open up all the other channels, you see the same thing, and you'll see that there's very, very little spread into all those other UV channels. Now, what we do in the spread matrix is we open up all the channels to develop it so we can see the cross laser problems associated with that too. But for each laser line, if you look at every color on it and then look for the spill into every addition, or the spread into every additional detector on that, you'll come up with a large Gaussian curve, which starts at the 395, gets larger and peaks in around the 600s, and then as you go into the 700s and 800s, it falls off and becomes very small again. So each laser line has that story, and you'll only see it if you take colors from every detector for every detector you have on the instrument, open them all up, and one by one check them, and you'll see that in terms of how we build a spread matrix. I think you can move on.
Thank you very much.
Yeah.
Yeah. Thank you. So the next question, and this one, Rui, will direct to you, is someone is asking how to properly set up their instrument settings on a TN-TV, and how does this impact their resolution?
In a what? Sorry, I didn't get that.
Right. So the question was how to properly set up their instrument settings, and how does this impact resolution in regards to TN-TV?
Right. Okay. EMT. Right. So it's so talking about let's say conventional flow cytometer with PMTs, and this is a very there's a lot of discussion every time we talk about setting up an instrument. It's very important to understand that, and usually when we say we need to have the instrument performing well, What it means is that we want to make sure that each channel is set up in such a way that we have maximum resolution from that channel, from that PMT. And what this means is that, so, PMTs have intrinsic electronic noise. And if we just use the default gains, or if we use a very low gain, we risk that the signal that we're trying to measure, both alpha fluorescence, which would be the background cell fluorescence, and the actual fluorescence that we're trying to measure, we risk that we won't be able to pull those apart because the signal is just basically overwhelmed with electronic noise. So what we do is we increase the voltage of that PMT in such a way that we increase the background fluorescence and the fluorescence, so that the electronic noise, which is not amplified because it's intrinsic noise of the PMT, stays the same, and hopefully becomes negligible compared to what we're trying to measure. So that's the whole objective of setting up the PMT voltages. We just want to increase them in such a way that the electronic noise is negligible. Now the question would be, well, why don't we just crank all the voltages of PMTs and just measure it like this? Because like that we assure that we're always far away from electronic noise. And that could be a way to do things. The only issue there is that if we use voltages that are too high, we risk that our signal will be off scale, right? And we lose a bit of that dynamic range if the voltages are too high. So the strategy here is typically to increase the voltages so that we reach maximum resolution. And maximum resolution is obtained when the electronic noise becomes negligible. And after that, if you continue increasing the PMT, you won't improve resolution, right? You'll just start losing that dynamic range. So again, the strategy is increase it until you reach maximum resolution and use that minimum voltage that will give you maximum resolution. So if you do that to all of the channels that you're going to use in your instrument, then you're all set. You shouldn't have to change your voltages if the instrument is performing the same way every day. So that would be the strategy. And then you can start titrating your antibodies. You can start validating your panel design, and eventually, it may take days to weeks to months to optimize everything. Once you do that, you can start running your samples. Of course, it depends on the size of the panel, right? But again, going back to the instrument, the most important thing is to ensure that you have the minimum voltage that gives you maximum resolution.
Thank you very much. Yolanda, this next question is going to be directed towards you. An audience member is asking if they need to run compensation controls every time they run an experiment, or for how long can they reuse the same compensation controls?
So that is a good question because it does come up regularly. And there are certainly people who would use their matrix or even compensation controls that they've stained for multiple days after another. And it works for some, but I would be very cautious doing that because it presupposes that nothing changes in your experiment or on the instrument between those days that you're doing that. So you have to use the exact same reagents and fluorochromes, obviously, throughout. The reagents have to be stable, which is not a given. The instrument settings have to be identical, and you have to be sure that the instrument performance is identical from day to day. And so everything has to be very narrowly controlled. And again, while that does work for some people in some settings, I would be very cautious doing that. And in most cases, preparing compensation controls is not the big problem. It's not the hurdle in your experiment. It's the biological samples that are harder to work with, right? So especially if you're using beads, there should be no reason not to stain compensation controls fresh together with your experimental samples every day. I wanted to add something, since we're talking about compensation controls. I wanted to add something to what Uri said earlier, that of course the compensation controls always have to be brighter than your experimental sample for any given fluorochrome. But overall, brighter is not always better. And yes, it has to be brighter than your cells that you're interrogating. But you don't want to push the signal all the way against the right axis either. So be mindful of the linearity of your PMTs. That is, in the upper decade, you kind of lose the linearity. So you don't want to push everything, to the maximum intensity. You want to be brighter, but not full throttle. So.
Yeah, that's so. I'd like to, just emphasize the, this thing of wanting to use always, the same, or to reuse the compensation controls. Like Yolanda said, you have to assume that not only the reagents are stable, are always the same, but you also have to assume that the instrument is always the same, and that's so difficult to obtain, even in an almost perfect world. But definitely, if you're using tandem dyes, which you typically will be, it's just impossible to guarantee that reagent will be the same that when you used it a week ago or a few weeks ago. So if you're using tandem dyes, just forget it. Like Yolanda said, it's not the most difficult parts, right? In running even a big panel, just run single stain controls every day that you run your experiments. So fresh. I think that's extremely important, like Yolanda said.
All right, thank you. This next question is directed towards Bob. Can you comment on the new Cytometry fluoros?
Okay, I'd be glad to. It's if we look at our history of dye development and fluorochrome development over the last 40 years, it took us a long time to get to 20, and I think with the innovation that came out of Sirigen, it was just an amazing way for us to really look at polymer technologies, and I think we all know in our field what it's done for us. I think today there are a number of there's several companies out there that are developing new dyes with newer technologies. These are scaffold technologies, and Cytometry is certainly one that has developed a whole series of them. I saw they're approaching 20. I think I saw just the other day. So I think that these are very exciting dyes for us all to look at and to evaluate, and where they fit out there in our field. I think it's still up in the air in terms of where we are with the dyes that we have in our portfolios and how we're using them. But what I can say is that the scaffold technologies that are coming out look to be really tuning dyes into specific channels that appear to be very good in lowering cross laser excitations and things like that, which again, in all of our work, whether or not it's conventional flow or spectral, I think is just going to add a new dimension to where we are and allow us not just to get higher and higher in plexing, because I think that's still just a very small percentage of the total individuals in the world that are working with that. But I think that a lot of these new dyes are going to perhaps in many ways, as we have with Sirigen technologies, replace some of those dyes and made some of the dyes in specific channels even better. So I think these new introductions are just of great value to our general field.
Thank you. Thank you very much. Does anybody else have any thoughts that they'd like?
Maybe I would just add there that we're always excited to hear about new dyes. These new Cytometry dyes are still they're still so new that there's a lot of testing in the field to be done yet. So they're very promising. We're hoping that, the fact that we can tweak the brightness. So this is a novelty that didn't really exist before. So with the same fluorochrome, if you can tweak the brightness, that probably will help a lot in terms of panel design. But again, it's still too early to say how well it will work, and even in terms of stability and things like that. So I don't know. We're just looking forward to seeing those guys and actually testing them. And we've purchased them already to test, but we haven't done it. So. Yeah.
So our last question for today, that an audience member has asked, that I'd like that I'll there, and I will decide whoever would like to address it, please do so. Is what is your preferred method to assess compensation accuracy? The use of N-by-N plots, or do you have automated strategies that you recommend?
Mm-hmm. So who starts?
Well, you're already nodding your head, so good.
Exactly. Well, so again, assessing, how correct your composition is relatively easy if you think of that the medians, when you're correcting the medians of the negative and the positive population in the secondary channels needs to match, right? So there are many ways to do this. Some people just look at it by eye. I think that's a bit dangerous because sometimes things are a bit offset, and you might think that they're not correctly compensated. And we've seen situations where, well, actually the negatives are, let's say, lymphocytes, and the positives are monocytes. So they have more autofluorescence in this secondary channel. So they are going to be a bit offset. And again, that's one of the dangers of then trying to tweak it by eye. But having said that, so typically you would see the medians match. We have one thing that we've developed in collaboration with a colleague of ours is what we're calling the secondary stain index, where basically we measure the stain index of the negative and positive population in the secondary channel, and this allows us to see how offset the populations are, and if it's more than 2 standard deviations away, then we'll deem it as okay. This is not well compensated. If it's within 2 standard deviations, then we just leave it like that. Even though it may seem a bit over or undercompensated, it's still statistically within what you would expect medians to match. So this is the typical way we do it, and it's very easy to set it up. Again, you can visualize it in an N-by-N plots, or you can simply set the gates and just have the software calculate all these secondary stain indices, and it will tell you immediately if everything is well compensated or not. So this is how we do it.
All right, thank you very much. We've reached the end of our time today for this Ask the Expert panel. I would like to say a special thank you to Yolanda Mahnke of FlowKnowHow LLC and Dr. Rui Gardner of the Memorial Sloan Kettering Cancer Center. I'd also like to thank Bob Balderas and Dr. Mirko Corselli of BD Biosciences for joining us today. For more educational resources for flow cytometry panel design, you can visit the flow cytometry panel design education page located on bdbiosciences.com. Broadcast of this on demand. The broadcast of this session will be available on demand at www.currentprotocols.com in the webinars section of the resources. Thank you for joining us today.